Hi everyone, welcome back to class. In today's class, what we want to discuss is catabolite repression. We want to know all about catabolite repression. What is catabolite repression? What is the importance of this repression? Why do we need it? How does it work? That is all we want to talk about today. And I promise you, it's going to be an interesting discussion today. What is catabolite repression all about? Catabolite repression is a regulatory mechanism, and its work is to inhibit the expressions of genes that are involved in the metabolism of alternative carbon sources when a preferred carbon source is present. Let me go over that again. It's a regulatory mechanism. It's a regulatory mechanism that helps in inhibiting the expressions of genes that are involved in the metabolism of alternative carbon sources. When a preferred carbon source is present, and it is mainly found in prokaryotes. Now let's try and break it down. Let's say in a bacterial medium, let's say we have in this medium we have bacteria. I'm representing it with B. You have bacteria, you have bacteria, we have bacteria. Now also here, let's say we have glucose. That is G. You have glucose, we have glucose, we have lactose, we have lactose. Now, let's go back to our definition. We said this repression type, it will help us to inhibit the expressions of genes that are involved in breaking and metabolizing alternative carbon source. Meaning that here, the bacteria will favor the uptake of glucose than lactose, because glucose can readily be absorbed by them and make use of it to further their metabolism. But if it were to be lactose, lactose will still need to be broken down before they will get the glucose and before they now start working on glucose itself. So the least one, the most convenient one they can go for is glucose that is there. Now let's try and expand it. Let's say this line is our bacterial cell. This is the inside of the bacteria. This is the outer part of the bacteria. And we said here we have lactose, we have lactose, we have lactose, we have glucose also. We have glucose also. Now, what will happen in catabolite repression is that there will be uptake of this glucose into the cell with the help of glucose transporter. So glucose transporter will enhances the uptake of this glucose into the cell. So we have glucose. As soon as there is entry of glucose into the cell, there is dephosphorylation of this enzyme, thereby making this enzyme active. Now, after the activation of this phosphotransferase enzyme, the next thing is that the enzyme will now help in the inhibition of adenylate cyclase. So there is uptake of glucose into the cell by glucose transporter. So glucose transporter enhances the uptake of the glucose into the cell. And when this glucose enters into the cell, it helps to activate this enzyme, which it does that by dephosphorylating this enzyme. So after this enzyme is being activated, that is phosphotransferase enzyme, this enzyme will now help to inhibit this enzyme, which is adenylate. Now, let's talk about adenylate cyclase on its own. What is that it helps in producing cyclic AMP. So adenylate cyclase will give us cyclic AMP. Cyclic AMP. So now, this cyclic AMP, upon binding of this cyclic AMP to catabolite activator protein, CAP, catabolite activator protein, let's say CAP, and this CAP is catabolite activator protein. So after adenine cyclase, what it does is that it produces CAMP. Now, adenine cyclase produces cyclic AMP. Now, upon the production of this cyclic AMP, it binds with CAP, which is catabolite activator protein. And when these two binds, combination of them can activate, they come to the promoter of the lycopene. This is our lycopene. It now binds to this promoter. So we'll be having AMP, then we have CAP. So remember, the combination of this binds with this, which upon binding, like, they activate this promoter. And as soon as this promoter is being activated, then RNA polymerase can start work, and there will be expressions of these genes. And when there are expressions of this gene, it will give us beta galactosidase. Now, beta galactosidase helps in breaking down lactose. But yes, since the cell has prevented the uptake of lactose, it is making use of glucose. So that means we did not need beta galactosidase to be produced. We don't need the expression of this particular gene. We don't need this gene to be expressed. So since if we would not be needing this to be expressed, that means we will find a way of preventing the binding of this cyclic AMP and CAP to the promoter. Now, what will now happen is catabolite repression is that upon entry of glucose into the cell with the help of glucose transporter, which leads to the activation of phosphotransferase by dephosphorylation, this enzyme phosphotransferase will now inhibit this enzyme adenate cyclase. And remember, adenate cyclase is the one that produces this CAMP. So when we inhibit this enzyme, there will not be production of this. So if there is no production of this, there is no how cyclic AMP and CAP will now come and activate the promoter here. So that means we are shutting down this lycopene. We are shutting it down. So telling the cell that we have sufficient glucose, continue to be using glucose. So now let's link it to our definition. We said it is a regulatory mechanism that will help in inhibiting the expressions of genes. These are the genes, the LAC-Z, LAC-Y, and LAC-A. These are the genes. So catabolite repression will help us to inhibit the expressions of this gene. And when these genes is expressed, their products, the products that we get from there will help in breaking lactose, which is other carbon source. But in this case, we don't want it to be broke. We don't need it because we've already prevented it. So now catabolite repression, we inhibit the expressions of this gene, and we do that by inhibiting adenate cyclase. So when adenate cyclase is inhibited, there will not be production of cyclic AMP. And when when cyclic AMP is not being produced, there is no how cyclic AMP will now join with CAP and activate this promoter. And so far this promoter remains unactivated, this system will not work. So this system is closed down like that. Then the cell will now be making use of glucose that is present. Now, another thing that this enzyme helps us to do is that it prevents the entry of lactose into the cell. We have permease here, permease that are enhancing the entrance of lactose. But upon activation of this enzyme, phosphotransferase enzyme. It helps to beat the entry of lactose. It's indeed the entry of lactose in the cell, and also it's now it increases the entry of glucose into the cell. So we have more glucose entering, we have more glucose entering into the cell, we have more glucose entering into the cell, which will now be readily used by the bacteria for their metabolism. We have more glucose that enters into the cell and which the bacteria will make use of. I hope you understand everything about catabolite repression by now. By this, we've come to the end of today's class, which is catabolite repression. See you next time. Bye bye.